Hello everyone, welcome to the new session of a chapter called Biomolecules. So in this session, we are going to study about the past year NEET question. This is the second part. In the first part, we have already discussed about some of the questions which came in the NEET examination. Okay, so here in the second part also, we are going to some of the questions which came to the NEET examination. The first question. So here it is the match the following type of question. In the column 1, it is given inhibitor of catalytic activity. The second, possesses peptide bond. Third, cell wall material in fungi and secondary metabolite. In the second column, it is given ricin, malonate, chitin and collagen fiber, or we can say collagen. So, inhibitor of catalytic activity. So, malonate, if you remember, succinate dehydrogenase. It is an enzyme and its inhibitor is, or we can say a catalytic inhibitor. It competes for the substrate. It competes with substrate to get attached with this particular enzyme. So when this malonate, which is totally resemble to this succinate, it get attached to the succinate dehydrogenase. So at that particular active sites of this succinate dehydrogenase, substrate could not get attached. And it inhibits the catalytic activity of succinate dehydrogenase. So the first match for the inhibitor of catalytic activity is malonate. It is given in the NCERT. So remember this. Possesses peptide bond. So protein is having peptide bond. So here which is protein? Collagen. It is an animal protein. Okay. Most abundant animal protein. So this is peptide bond is present in the collagen protein. So this is the match. Cell wall material in fungi. This is very, very important. Chitin. Cell wall material is in fungi is made up of chitin. Chitin is also present in the exoskeleton of insects and crustaceans. Or we can say simple exoskeleton of the arthropods. Okay, so remember this. Chitin is found in cell wall of fungi as well as the exoskeleton of insects and crustaceans. Secondary metabolite, ricin. So this is also a very, very important. So our correct match is A, 2, B, 4, C, 3 and D, 1. Okay. Moving forward. So this is our correct match. Next question. Match the list 1 and with list 2. So what they given in the list 1? Protein. B, unsaturated fatty acid. Nucleic acid. Polysaccharide. In the list 2, it is given carbon-carbon double bond. Phosphodiester bond. Glycosidic bond. And peptide bond. So protein, we all know that the protein is formed by the formation of polypeptides. And polypeptides are what? They are the chains of amino acid. Like this. And amino acids are linked with a bond which is called as peptide bond. So very important. Protein is having peptide bond. unsaturated fatty acid. So remember saturated fatty acid and unsaturated fatty acid. In the case of unsaturated fatty acid, you will have CC double bond in the carbon chain. Whereas in the saturated fatty acid, all the bonds got saturated and it got its name saturated fatty acid. So, okay, so unsaturated fatty acid CC double bond is there. Got it? Next, nucleic acid, very important, phosphodiester bond. So we are having ribose sugar, we are having this nitrogenous base, that could be purine and pyrimidine, anything, and here is the glycosidic bond, and here we are having, this is the 5th carbon, and here we are having phosphate group. I'm just drawing a rough sketch, phosphate group. So these are called as phosphodiester bond. Okay, so phosphodiester bond is present in nucleic acid. Polysaccharide, glycosidic bond, whether you study about the starch, whether you study about the cellulose, whether you study about the disaccharide, trisaccharide, anything, you will have glycosidic bond, right? Next, following are the statement with reference to the lipid. So these are all the 5 statements that is concerned for the lipid. Okay, so let us read about one by one these statements. Lipid having only single bond and are called unsaturated fatty acid. Lecithin is a phospholipid. So if you remember, the plasma membrane is having a type of a lipid which is called as phospholipid. And this lipid example is lecithin. So lecithin is one of the phospholipid. Trihydroxypropane is glycerol. So here we are having CH2OH, CHOH, CH2OH. So this is propane, meth, eth, prop, and trihydroxy because it is having 3 hydroxyl group. So trihydroxypropane. Palmitic acid has 20 carbon atoms including carbonyl carbon, and arachidonic acid is having 16 carbon atoms. So if you read the NCERT, okay, you will get that palmitic acid is having 16 carbon atom, whereas arachidonic acid is having 20 carbon atom. Lipid is only having single bonds are called unsaturated fatty acid. This is incorrect. So here we are having B and C statement as only correct. Only B and C statement are correct. Next question. Read the following statement on the lipid and find out the correct set of statements. Lecithin is found in the plasma membrane and is called glycolipids. Saturated fatty acid possesses one or more carbon-carbon double bonds. Gingerly oil has a lower melting point and hence remain as oil in winter. Lipids are generally insoluble in water but soluble in some organic solvent. When fatty acid is esterified with glycerol, monoglycerides are formed. So lecithin is found in the plasma membrane, which is a phospholipid. Saturated fatty acid never possesses the carbon-carbon double bond. So these two statements are incorrect. Gingerly oil has a lower melting point, yes, and it remains as oil even in the winter. This is one of the example of the oil which remains oil in winter when the temperature is so low, right? Lipids are generally insoluble in water, yes, but soluble in some organic solvent. This is also a correct statement. And when one fatty acid esterified with glycerol, monoglycerides are formed. This is also incorrect. So our correct statement are third, fourth and fifth statement. Next question. Given below are the two statements. A protein is imagined as a line, the left and represented by the first amino acid, that is the C terminal, and the right and represented by the last amino acid, that is the N terminal. So if this is a polypeptide which is made up of, here this is the first amino acid and here it is the last amino acid. So if I say that this polypeptide is made up of 110 amino acid, this is a residue of a polypeptide which is made up of 110 amino acid, then this first one will have a structure like this: alpha carbon, COOH, NH2 group, H, and suppose this is a alkyl group. So here you can see this particular end is free, which is called as N-terminal, and here at the last amino acid, what is free? NH2, alpha carbon, COOH, H, and here R2. That can be a different R. So here this 99th amino acid will have here COOH group, and this hydrogen OH and this one of the hydrogen form a bond, remove a water and form a bond. So this is not free. What is free here? COOH group is free at the C terminal. This is called as C terminal. So the left end is having N terminal free and the right end of the polypeptide will have C terminal free. So this statement is incorrect. Adult human hemoglobin consists of 4 subunits, 2 subunits of alpha type and 2 subunits of beta type. So if you remember, this is also given in the NCERT. If you remember the composition of hemoglobin, then they are having 2 alpha and 2 beta subunit. And total they are having 4 subunits. Okay. So this statement is totally correct. So we are having the 4 option. Statement 1 and statement 2 are true, means both are correct. Statement 1 and statement 2 are false, mean both are false. Statement 1 is true and statement 2 is false, which is incorrect. But statement 1 is false and statement 2 is true, which is the correct. Statement 1 is false and statement 2 is true. Okay. Next question. Given below are the 2 statements. Again, statement-based question. And nowadays, statement-based questions are very common in the NEET examination. There are different types of statement question, like statement 1, statement 2. There can be a multiple statement types. And it can be asked which one is correct. They can also give a combination of multiple statement and they will ask that which of the combinations are correct. Right? So be carefully. First of all, read the statement very, very carefully. Okay. Low temperature preserve the enzymes in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. So what they are saying, enzyme when stored at lower temperature, lower temperature. They are inactive, but they are stored. What happened at the higher level? At the higher temperature, the protein get denatured because at that particular point, the peptide bond get broken and that polypeptide get denatured, or we can say the protein get denatured. So this enzyme act at one particular temperature and that is called as optimum temperature for its activity. But below that, it can be present in the inactive state temporarily, but when it reaches again to the optimum temperature, it again behaves as a active enzyme. But when its temperature rises, it gets denatured. When the inhibitor closely resembles the substrate in its molecular structure, it inhibits the activity of the enzyme. It is known as competitive inhibitor. So if you remember one of the example which is given in the NCERT, succinate dehydrogenase. Succinate is there. So this is a substrate and this is the enzyme. Now there is an inhibitor, malonate. It totally resembles to this succinate. So what happened when malonate comes, it join with the succinate, and do not allow the succinate to join with the succinate dehydrogenase and forms the product. So here it is doing what? It is competing. Malonate is competing with substrate to join with the succinate dehydrogenase. And why it is joining with the succinate dehydrogenase? Because it is closely resembles to the substrate. Okay, so both the statements are correct. So both statement 1 and statement 2 are true or correct. Next question. Which one of the following biomolecule is correctly characterized? Lecithin, a phosphorylated glyceride found in the cell membrane. Yes, it is a glyceride molecule which is phosphorylated. Okay. Palmitic acid, an unsaturated fatty acid with 18 carbon atom. It is 16 carbon atom. Adenylic acid, adenosine with glucose phosphate molecule. And alanine amino acid contain an amino group and an acidic group anywhere in the molecule. Alanine amino acid contain an amino group and an acidic group anywhere in the molecule. This is incorrect statement. So our correct statement is only lecithin phosphorylated glyceride, okay, which is present in the cell membrane. Next question. Spoilage of oil can be detected by which of the fatty acid? Oleic acid, linolenic acid and linoleic acid. Linolenic and linoleic acid and erucic acid. So this erucic acid is an acid which get spoiled, and it can be detected by its foul smell. These oleic acid and linolenic acid and linoleic acid do not get detected or get spoiled. Okay. So erucic acid is the answer. Next question. Essential amino acid is, which of the following is the essential? So if you remember, essential and non-essential amino acid, phenylalanine is the essential one. Essential amino acid. So we require it as a dietary component. Aspartic acid, glycine and serine, these are all non-essential because this can be synthesized in our body. So the correct is phenylalanine. Next question. Which of the following statements regarding enzyme inhibition is correct? So this, now you can see this, many question has been asked on the inhibition of the enzyme. So read about this from the NCERT, and whatever the statement is given here, the question will come from those only. Okay. So understand it and read this particular statement and try to find the correct answer. Competitive inhibition is seen when a substrate competes with an enzyme for binding to the inhibitor protein. So competitive inhibition is seen when substrate competes with an enzyme for binding to an inhibitor protein. No, substrate compete with the inhibitor protein to bind with the enzyme. Are you getting? So there is a substrate, there is an inhibitor, both are structurally same. And here we are having enzyme. So this substrate and this inhibitor will compete each other to get bind with the enzyme. So what it written here is substrate competes with enzyme for binding to an inhibitor. Substrate is never going to bind with the inhibitor. It is going to bind with the enzyme. So this is incorrect statement. Competitive inhibition is seen when the substrate and the inhibitor competes for the enzyme active sites on the enzyme. This is the correct statement. This substrate and inhibitor molecule are competing with each other to go and bind with the enzyme's active sites. There are some sites or the crevices in the structure of enzyme where the active sites are present, and the substrate goes and bind to that active sites. Now there is one inhibitor also. This inhibitor is structurally same to the substrate. So it also compete with the substrate to get bind with the enzyme's active sites. Okay. Non-competitive inhibition of an enzyme can be overcome by adding large amount of substrate. Non-competitive inhibition. There is no competition. Okay. Of an enzyme and can be overcome by the adding large amount of substrate. Next question. Non-competitive inhibitors often bind to the enzyme irreversibly. So correct is, both the statement are incorrect. Competitive inhibition is seen when the substrate and the inhibitor compete for the active sites on the enzyme. This is the correct statement. Next question. 3 of the following statement about the enzymes are correct and 1 is wrong. Which one is wrong? So only one statement is wrong and you have to find that. Enzyme require optimum pH for its maximal activity. Yes. Temperature and pH are the external factor on which the enzymatic activity depends on. So they require optimum pH. Enzymes are denatured at high temperature, but in certain exceptional organisms, they are effective even at temperature 80 to minus 90 degrees centigrade. Yes, in some of the thermophilic bacteria, their enzymes are active even such a harsh condition like 80 degrees centigrade or minus 90 degrees centigrade, right? Enzymes are highly specific. Most enzymes proteins are protein. Most enzymes are protein, but some are lipids. So this enzyme are highly specific. This is also correct. But most enzymes are protein, but some enzymes are lipid. This statement is incorrect. Lipids are not enzymes, right? So this is incorrect. So fourth statement, most enzymes are proteins, but some are lipid. This is incorrect statement. Next, which one of the following is a non-reducing carbohydrate? Non-reducing carbohydrate means absence of CHO and ketone group. So these are the two groups which get reduced. and the one which is not having these two groups are called as non-reducing carbohydrate. So maltose is having those group, lactose and ribulose 5-phosphate. Sucrose is not having a CHO or ketone group, aldehyde or ketone group. So sucrose is a non-reducing carbohydrate. Carbohydrates are commonly found as a starch in plant storage organ. Yes, carbohydrate. Starch is a type of carbohydrate, and that is a storage carbohydrate in the case of plants. Which of the following 5 properties of starch from 1 to 5 make it useful as a storage material? Very important. 1 to 5 statement, and out of these 1 to 5, the statement which says that this starch is useful as a storage material. Okay, so easily translocated. It has nothing to do with storage. Chemically non-reactive. So something when it is stored, it should not be reactive, right? So chemically non-reactive is the correct statement for the storage material. Easily digested by animals. This is also nothing to do with the storage. Osmotically inactive. Osmotically inactive. Yes, it should be inactive, means storage. So there should not be it should not be move out osmotically, right? So this is also the correct regarding the storage material. Synthesized during photosynthesis. Glucose is synthesized, and when it is stored in the form of starch. It is converted to starch. So again, this is incorrect. So 2 and 4 is the correct statement, right? Next question. The catalytic efficiency of the two different enzymes can be compared by formation of the product, pH optimum value, K value, and the molecular size of the enzyme. So if you remember the graph, substrate, and here we are having the velocity. The catalytic efficiency of the two different enzymes can be compared by, so formation of the product, in both the case of two different enzymes, the product will be different. So how can you measure the catalytic efficiency? pH of optimum value. In both the cases, the two different enzymes, so both will have the optimum value different. Molecular size of the enzyme. This also doesn't gives the efficiency of the enzyme activity. Molecular size. Some enzymes are smaller, low molecular weight, and some will have the high molecular weight. But its enzyme activity can be larger, right? And this large molecular weight enzyme can have low efficiency of its activity. So this K value, formation of the product, or we can say the velocity of the enzymic activity. Okay, this gives you the catalytic efficiency of the two different enzymes. You can compare the catalytic activity of the two different enzymes by this characteristics of the enzyme. Next, enzyme having slightly different molecular structure but performing identical activity. Holoenzyme, isoenzyme, apoenzyme, and coenzyme. Now, holoenzyme is what? Holoenzyme is equal to apoenzyme plus coenzyme. So coenzyme can be prosthetic group, can be cofactor, can be metalloin. So these are the different types of coenzyme. So, and this apoenzyme is totally a protein part. a protein part. And this is the holoenzyme. So now going back to the statement, enzyme having slightly different. Now there are two different enzymes. They are talking about two different enzymes. And this particular enzyme is having a different molecular structure. Some part is different, but both are going for the same activity, performing the identical activity. So isoenzyme, because they are having same activity, identical activity. But if you see this holoenzyme, apoenzyme, coenzyme, this is nothing to do with the activity, identical activity, right? So the isoenzyme is the correct answer. Next question. Which of the following is the least likely to be involved in the stabilizing the three-dimensional folding of the protein? Hydrogen bond, electrostatic interaction, hydrophobic interaction, and ester bond. So if you remember the primary, secondary, tertiary, and quaternary structure of the protein, we are having peptide bond, we are having covalent bond, we are having ionic bonds, we are having van der Waals forces, we are having hydrophobic interaction, hydrophobic interactions. These two are the weak interaction, but they are very important weak interactions. And we are having very important hydrogen bonding. Hydrogen bonding is present in the secondary structure. It is present in the secondary. It is present in the tertiary. It is present in the quaternary structure. But peptide bond is simply present in the primary structure. So these are the different types of bonds involved in the formation of protein structure. No ester bond is present in the formation of any type of conformation of the protein. Any type, that is primary, secondary, tertiary, or quaternary structure. Okay. So ester bond is not involved in stabilizing the three-dimensional folding of the protein. Okay, so the correct answer is ester bond. Next question. Which is not consistent with the double helical structure of the DNA? Not consistent. AT is equal to CG. Means A always combined with T. Okay, compare form the bond, hydrogen bond with the T, and C with the G. 3 bonds. Density of DNA decreases on heating. A plus T upon C plus G is not constant. Both A and B. So which is not consistent in the helical structure of DNA? So A plus T upon C plus G. So I'm just taking one example. If A is 20% in a DNA sample, okay, then T will also be 20% because A always pair with T, right? Here it should be triple bond. Okay, so this is correct. Coming back to this. So what will be then C and G percentage? So total is 100%. If we add 20 plus 20, then it will be 40%. A and T combinedly, it will be 40%. Okay, so what will be the percentage of C and G? What will be the percentage of C and G? So 100% minus 40%. 100% minus 40% will be what? 60%. 60%. Right? And C is equal to G. So both will be have 30% and 30%. So now according to this one, A plus T upon C, if we add 20 plus 20 is A plus T will be upon 60 plus 60. It is constant? No, it is not a constant. It is not a constant. Sometime the percentage of adenine is different. Sometime percentage of G is different. It is not a constant. Okay, so which is not consistent with the double helical structure of the DNA. So this is not a consistent, but yes, adenine always pair with T and cytosine always pair with G. This is consistent. Density of DNA decreases on heating. Yes, when you heat, the DNA get denatured, so its density decreases, right? So this is the correct statement. Next, in double helix of the DNA, the two DNA strands are coiled around the common axis, coiled around each other, coiled differently, coiled over protein sheets. So where it is present, there is one common axis and DNA structure is like this. DNA structure is like this. Okay, so in double helix of DNA, the two strands are coiled around a common axis. They are not coiled around each other. They are not coiled differently or coiled over a protein sheet. These three are totally incorrect. So they are coiled around a common axis. Next question. Transition state structure of the substrate formed during an enzymatic reaction is transient and unstable, permanent and stable, transient but stable, permanent but unstable. So enzyme when combined with substrate, it form ES, but later it form product and substrate, and this is also a very for a short duration, and later what happened? Product and enzyme. Later what happened? Product is released and enzyme is again available for the next round of reaction. So here this ES, this is transient stage. This is transient stage and unstable. It is not a stable compound. It is unstable. So the first statement, transient and unstable, is the correct. Next question. The given diagram representation show one of the categories of a small molecular weight organic compound in the living tissue. Identify the category shown and one blank component X in it. So here we can see one blank component. So this is a sugar and this is X is what? With this X, it is a place of nitrogenous base. Nitrogenous base. And here you can see it is a hydroxyl group is attached here. Hydroxyl group is attached. So this is a ribose sugar. No phosphate group is attached here. No phosphate group is attached here. So this says when ribose or deoxyribose get attached with the nitrogenous base, it form nucleoside. Nucleoside. And what is it is here? Nitrogenous base. So nitrogenous base are so different. Purine, pyrimidine, okay, adenine, guanine, thymine, cytosine, and uracil. In case of RNA, instead of thymine, there is a presence of uracil. So nucleoside is the correct category of this particular structure, this whole structure. And this X is what? This is a nitrogenous base, and here we are having adenine, guanine, thymine, cytosine, and uracil. So here uracil is the nitrogenous base. So the correct is nucleoside and uracil. So this is all about for this session. We have discussed so many questions which have came in the past year NEET examination. Okay, so do prepare your study according or the exercises which you do. Find out the questions and try to solve those questions according which we have done here. It will help you in your study. So we will meet again with one more interesting session. Till then, thank you very much.